Hello, this presentation is on the function of pulmonary surfactant. This is by Arlene Perez through the College of Science, Grand Canyon University, Human Physiology 2. Surfactant is a surface-acting material that is responsible for lowering the surface tension of a fluid. It lines the epithelium of the alveoli in lungs. The purpose to surfactant is to decrease the surface tension on the alveolar membrane. The objective in this presentation is to understand the source of secretion of the pulmonary surfactant, the chemistry of surfactant, the substances that are necessary for the formation of surfactant, the function of surfactant, the mechanisms of surfactant, deficiencies with surfactant, and overproduction and dysregulation of surfactant proteins. The alveolar system in the lungs has the largest surface of the body that is exposed to the environment. It is made up of two types of epithelial cells, pneumocytes 1 and pneumocytes 2, also known as alveolar cells type 1 and alveolar cells type 2. Type 1 cells are flat cells with a few organelles and are not able to divide. They work as a thin barrier between the blood and air. They function as a cation transport, but also work as a signal to the type 2 cells. Type 2 cells are cells that take part in the clearance and repair by moving forward to areas that have been damaged. These cells are able to divide and can differentiate into type 1 cells. The main function of type 2 cells is the secretion of surfactant in the lungs. Surfactant is made up of a combination of different molecules. It is mainly made up of phospholipids and specific proteins. These proteins are surfactant protein, SPA, SPB, SPC, and SPD. SPA and SPD are part of a family of large multimeric proteins with collagenous lectin hybrid properties and are involved in host defense. They are water-soluble and bind lipids. SPA also organizes alveolar surfactant phospholipids. SPB is a protein of a different family that includes lipid-interacting polypeptides that have antibacterial and lytic properties. SPA, SPB, and SPD are also found in the gastrointestinal tract. SPC is the only one of the surfactant proteins that is only found in the lungs. It is a lipopeptide that interacts with the membrane with a fluid bilayer made up of di-polymaltoglycophosphochlorine, the main composition of surfactant. The type 2 alveolar epithelial cells are the main source of surfactant. Type 2 cells have lamellar inclusion bodies that are rich in phospholipids. This is a substance that is similar to surfactant. At one time, it was believed that surfactant was made in the lamellar bodies, but it has been shown that it is made in the endoplasmic reticulum and stored in the lamellar bodies before it is released to the surface of the alveoli. Before newly made phospholipids can be moved from where they were made to the lamellar bodies by phospholipid transfer proteins. Surfactant can be recycled. The alveolar type 2 cells that make up about 7% of the surface of the alveoli produce surfactant with dietary substrates. Surfactant is made in the endoplasmic reticulum of type 2 alveolar cells. It is then carried to the Golgi apparatus for modifications. Majority of the components of the surfactant get stored in the lamellar bodies till they get secreted into the liquid hypophase found in the alveoli through exocytosis. A lattice-like structure is formed by surfactant known as the surfactant film. Phospholipids are either incorporated or broken down by macrophages, or they get recycled back into the type 2 cells so they can be used again. There are three main functions of surfactant. The first is to increase the compliance of the lung. The second is to prevent fluid accumulation in the alveoli. And the third is to stabilize the size of the alveoli. To explain the first function, we need to understand that surfactant decreases the surface tension as well as lowers the tendency of the alveoli to collapse. Because of this, there is less force needed to expand the alveoli. As long as surfactant is present, it is easier to expand the alveoli or the compliance of the lungs increase. It also makes it easier to breathe, so there is not much work necessary when breathing. With the second function, if we understand that if the high surface tension of surfactant had not been present, it would collapse the liquid in the alveoli. This would bring in fluid from the interstitium into the alveoli, making the thickness of the liquid layer higher and more difficult for diffusion of gases. Since surfactant does not decrease the surface tension, it prevents this from happening. Laplace's law, which states that the pressure equals 2 times the tension divided by the radius, this helps to explain the third function of surfactant. The more the tension, or smaller radium, the higher the pressure. So if two alveoli have a similar size after inspiration, they would both be lined with fluid. As the surface tension of the fluid increases, pressure on the air in the alveoli would be equal. As the alveoli collapse during expiration, the density of surfactant increases, resulting in a decrease in the surface tension in both alveoli. As surfactant adjusts in the surface tension, it stabilizes the size of the alveoli. Surfactant molecules are molecules that lower surface tension between two materials, either a gas or a liquid. All surfactant molecules are made up of a hydrophilic or water-soluble head and a hydrophobic or oil-soluble tail, or they are molecules known as amphiphilic. Surfactant starts by grouping on the surface of the water to create a water-air interface through the hydrophilic head towards the water and the hydrophobic tail towards the end. This configuration results in a decrease in the surface tension. The hydrophilic part of the molecule line the alveoli and the hydrophobic tail towards the alveolar air. These phospholipid spreads throughout the alveoli to reduce the surface tension. Surfactant is critical for normal lung function in newborn and adult mammals. If there was not a proper amount of surfactant, the alveoli would collapse as one breathes out. This would cause compromise in gas exchange. The surfactant can be vulnerable to pulmonary toxicants. Damage to surfactant or impairment of its production can have an adverse effect on lung functions and can even be unsuitable with life. Deficiency of surfactant can cause a reduction in pulmonary compliance, causing an increase in the surface tension. This can cause a collapse of the alveoli at expiration. The total surface area for gas exchange and alveolar capillary diffusion capacity can also be reduced. This can lead to hypoxia and hypercapnia. Overproduction of surfactant can lead to damaged, impaired lung function. Pulmonary alveolar proteinosis PAP patients have difficulty breathing, have suffocant accumulation in the alveoli that does not allow the lungs to inflate, not allowing normal gas exchange. Acquired PAP can come from hematological cancer, drugs, inhaled particulate matter, and infections. Elevated suffragant levels suffer from dyspnea and reduced lung functions. Surfactant is a surface-acting material responsible for lowering the surface tension of a fluid. It lines the epithelium of the alveoli in the lungs and decreases the surface tension on the alveolar membrane. It is made up of two types of cells, type 1 alveolar cells and type 2 alveolar cells. It is the type 2 cells whose main function is to secrete surfactant in the lungs. Surfactant is made up of a combination of molecules, mainly phospholipids and specifically proteins. Surfactant is released in the surface of the alveoli and can be recycled. Three main functions of surfactant are the compliance of the lungs, prevention of fluid accumulation in the alveoli, and to stabilize the size of the alveoli. Surfactant molecules are amphiphilic, which results in a decrease in the surface tension. Deficiencies and overproduction of surfactant affect the surface tension and lead to damage and impair lung function. The following slides are references to this presentation. Thank you.